Okay, so welcome back to this next video in which we are discussing phospholipase C enzymes. Okay, so in the previous video what we did is we discussed what phospholipase C enzymes do. And remember, when we say phospholipase C enzymes, strictly speaking, we mean phosphoinositide-specific phospholipase C enzymes. And we saw that the main thing that they do is break down phosphatidylinositol 4,5-bisphosphate into diacylglycerol and IP3, okay, and that these two molecules have key signaling roles within the cell. Okay, we then saw all the different 13 genes which code for phospholipase C enzymes and how they can be divided into phospholipase C beta, phospholipase C gamma, phospholipase C delta, phospholipase C epsilon, phospholipase C zeta, and phospholipase C eta families. Okay, and we're going to go through these families now in turn. Okay, and we're going to start with the phospholipase C beta family. Now, what we've seen that is common for all of these families of phospholipase C enzymes is that they all have a catalytic domain, which is one of these tin barrel-like domains, which consists of the alternating beta strands and alpha helices, and the beta strands end up forming a beta sheet which is folded into a cylindrical shape in the center, and then the alpha helices are around the outside. Okay, and there are two portions of the primary structure of the polypeptide which contribute to making the tin barrel-like domain. There's the X box and the Y box, and these two regions will have the alternating beta strands and alpha helices, and both of them will contribute to making the tin barrel-like domain, as I've shown here. So here's the portion made up by the X box region, and here's the portion made up by the Y box region. And then there is a linker between them called the XY linker, and this is believed to play an auto-inhibitory role. It's believed to sit in the catalytic site here and block it. And in order to activate the catalytic domain, you need to remove that XY linker from the catalytic site so that the Pi45P2 molecules can insert their heads into that catalytic site and the hydrolysis can occur. Okay, right, so let's now turn our attention to the specific family of phospholipase C beta enzymes. Okay, so firstly, let me tell you about the localization of the different phospholipase C beta enzymes, okay, because the expression of phospholipase C beta enzymes is different, basically. So you have different phospholipase C beta enzymes expressed predominantly in different tissues. Okay, so let's do this. So phospholipase C beta 1 then, firstly. Beta 1 is mainly found in the cerebral cortex. Okay, so you find it in high expression in the neurons of the cerebral cortex, and it's going to be involved in signaling in those cells. Okay, phospholipase C beta 2, meanwhile, you mainly find that in hematopoietic cells. Okay, so cells of the blood, white blood cells, red blood cells, platelets, these are all hematopoietic cells. Okay, so phospholipase C beta 2 is mainly expressed in hematopoietic cells. Phospholipase C beta 3 has a very broad expression. It's found all over the place, basically. Okay, so loads of different cell types will have phospholipase C beta 3 expressed. And finally, phospholipase C beta 4, this one is mainly found in the cerebellum. Okay, so the smaller portion of the brain that sits underneath the cerebral hemispheres. In the cells of that, you're going to find phospholipase C beta 4 expressed highly. Okay, however, these four members, they all have the same sort of broad structural features, and many of them are regulated in the same way. So, for instance, these are the ones which are going to be activated by alpha Q/11 family alpha subunits of heterotrimeric G proteins, okay, which we'll spend some time discussing in a moment. Okay, we'll also see how certain of them, particularly phospholipase C beta 2, can also be activated by the beta gamma subunits of heterotrimeric G proteins. Okay, right. So firstly, what I want to discuss is the structural features of phospholipase C beta enzymes. What structural features do they all share? Well, we know they're going to have one of these tin barrel-like domains as the catalytic domain, but what are the other domains that are important in the phospholipase C beta enzymes? So let's turn our attention to that now. Okay, so let's firstly draw the amino terminus of the protein here. And it's important to remember that all of these enzymes are one protein. Okay, they're not multi-protein complexes. It is a single protein that makes up the entire phospholipase C enzyme. Okay, right. So the first important domain that's worth noting, that's the most N-terminal of the important domains, is what's called a plextrin homology domain, which I'll just abbreviate as a pH domain. Okay, so we'll highlight the plextrin homology domain up in orange here. Okay, so I'll write out its name in full over here. So this is what's called a plextrin homology domain. Okay, and it's a domain that you find in many different proteins. Okay, and it's called a plextrin homology domain because it was, well, the first domain of this type was found in a protein called plextrin, which is important in platelets. Okay, now plextrin homology domains are usually important in binding to phospholipids within the phospholipid bilayers. Okay, so these usually attach proteins to the phospholipid bilayer. Okay, so you might be thinking, well, is that what this is going to be involved in doing in this phospholipase C beta enzyme? And the answer is no. In phospholipase C beta enzymes, we don't believe this plextrin homology domain is important for binding to phospholipids. Okay, so it's important to note that plextrin homology domains are not identical between different proteins, i.e. if you look at the actual sequence of amino acids in this plextrin homology domain and compare it to a plextrin homology domain in another protein, it won't be identical. Okay, even if you compare it between the different forms of phospholipase C beta, it won't be identical. Okay, and not all plextrin homology domains therefore actually successfully bind phospholipids. Okay, and these ones in phospholipase C beta enzymes, we don't think they successfully bind phospholipids, and we don't really know what the function of it is in phospholipase C beta enzymes. It may be redundant. Okay, right. Then, after the plextrin homology domain, you then have another domain which contains 4 EF-hand domains. Again, domains that are usually really important for doing a certain function, but which don't seem to do that normal function in these phospholipase C beta family enzymes. So these are representing 4 EF-hands, basically. Okay, and we'll colour these 4 EF-hands in red here. Okay, now what is an EF-hand then? Okay, so EF-hands generally occur in dimers, so really what we've got here is 2 dimers of EF-hands. And basically an EF-hand consists of just a little loop in the polypeptide like so. And as I say, they usually occur in dimers, so you'll usually have another one like this. Okay, and in this set of 4 EF-hand domains, what you'll have is 1 dimer, and then later on you'll have another dimer. And basically, usually, they are important for binding calcium. Okay, so usually a calcium ion can coordinate within the loop, and another calcium ion can coordinate in this loop here. Okay, so a famous example of a calcium binding protein which contains EF-hand dimers is calmodulin, a really important protein in calcium signaling. Okay, but in these phospholipase C beta enzymes, we do not think that the EF-hand domains bind calcium, but this is what they usually do. Okay, so I'll just stress that. Again, this is what EF-hands usually do, but in these phospholipase C beta enzymes, we don't think they do that. We don't think they're important for binding calcium at all. So again, like the plextrin homology domain, they seem as, you know, in other proteins they have a really important function. Here, they just seem to be there, basically, but they don't seem to be doing anything that's actually that important. Okay, so you've got these 4 EF-hand domains, but they don't bind calcium in phospholipase C beta enzymes. Okay, then what you've got is the catalytic domain here. So we'll draw the catalytic domain that we've studied previously. So here's our tin barrel-like domain. Okay, like so. Here's the catalytic site here where the PIP2 molecule will insert its head into. And it's important for me to note that you only have an opening to the catalytic site at one end of the tin barrel-like domain. Okay, so the tin barrel-like domain on the other side, down here, there isn't an opening there as well. It's blocked off there. Okay, portions of the protein block it off. So there's only a cavity at one side. So there's only one catalytic site for this. Okay, so here's the X box, and here's the Y box, and then there's that important XY linker, which will be obscuring the catalytic site, and therefore inhibiting the enzyme. Okay, right. So then, the next structure that we're going to have a look at is what's called the C2 domain. Okay, so this follows the Y box here. So after the Y box, what you have is a domain called a C2 domain. Okay, and again, C2 domains are usually extremely important in calcium signaling. They're usually extremely important for binding calcium, but in phospholipase C beta enzymes, and this is where phospholipase C beta enzymes are going to be different from some of the later types of phospholipase C enzymes that we look at. In phospholipase C beta enzymes, these C2 domains do not seem to be doing anything, basically. But in later phospholipase C enzymes, for instance, the phospholipase C delta enzymes, the C2 domain is going to bind calcium. Okay, right. So this is a C2 domain here. Now let's just have a little bit of a discussion of what a C2 domain is. So basically, a C2 domain is a beta sandwich structure. Okay, it's made of two beta sheets stacked on top of one another. So let me show this. So you'll have one beta sheet like so. Okay, and then you'll have another beta sheet underneath. Okay, so both of these beta sheets will consist of beta strands like so, forming a beta sheet. Okay, so there's one beta sheet, and then you can imagine that this will come up, and then you'd have another beta strand sheet like so. Okay, right, now it wouldn't really be that simple in reality, but this gets the basic principle across, that what you've got is two beta sheets stacked on top of one another, and that's what a beta sandwich is. Now, usually, these beta sandwich structures that you have within C2 domains are capable of binding calcium in these loops between beta strands. Okay, so for instance, in this loop here, we might be able to coordinate a calcium ion. Now, not all of these loops will be able to coordinate calcium ions, but some of them are capable of coordinating calcium ions usually. So that's usually how C2 domains work, that they are, again, a calcium binding domain, and when calcium binds to them, it causes some functional change in the protein that they are within. Okay, so we'll see how those are very important in phospholipase C delta enzymes for binding to calcium and getting the enzyme to respond to calcium. But in the case of phospholipase C beta enzymes, we don't think they're important at all. Okay, so we don't think they bind calcium, and therefore, again, it's another one of these domains that seems useless in the phospholipase C beta enzymes. Okay, right, now we've seen loads of unimportant domains that could be important in other proteins but aren't important in the phospholipase C beta enzymes. Now it's time to actually see some domains that are important for the function of the phospholipase C beta enzyme. Okay, so following this C2 domain, what you have is a region known as the H-alpha-1/H-alpha-2, okay, which I'll have like so. And we'll colour in this H-alpha-1/H-alpha-2 in green. Okay, and this region is what's called a helix-turn-helix motif. So H-alpha-1/H-alpha-2 is called a helix-turn-helix. Now let me explain what that means. It basically means that you're going to have an alpha helix followed by a turn, followed by a helix. I know, how obvious. Right, so what does this actually mean? So basically what you're going to have is an alpha helix, so a spring-like coil of the polypeptide. Then it's going to suddenly have this turn, and then you're going to have a new alpha helix form. That's all this means, basically. And the first alpha helix is known as the H-alpha-1 helix. Now, in our picture, this would be the first alpha helix, because you'd have the C2 domain here. Okay, so that will be called helix alpha-1, so alpha helix 1. And then you'll have alpha helix 2 here. Okay, so that's what the H-alpha-1/H-alpha-2 domain is. It's a helix-turn-helix motif. Okay, and that's actually going to be really important in the mechanism by which alpha-Q/11 subunits of heterotrimeric G proteins activate these phospholipase C beta enzymes. Okay, so just to stress, that's the turn there. Okay, right. So then the final important domain of the phospholipase C beta enzymes is what's known as a coiled coil domain, okay, which I'll put here. And initially I'm just going to put it as a box so that it's simple, but before I explain what a coil-coil domain is, and then after I've explained what a coil-coil domain is, I'll show this slightly differently. Okay, so there's our coil-coil domain, and we'll have the coil-coil domain in pink here. Okay, right, so what is a coiled-coil domain? Okay, well, coil-coil domain basically consists of multiple alpha helices coiled around one another. So an alpha helix is one of these spring-like structures. It's where the polypeptide folds into a spring-like structure like so. Okay, now to turn an alpha helix into a coil-coil domain, what you need is multiple alpha helices. And in this coil-coil domain that we have in the phospholipase C beta family enzymes, you're actually going to take 3 alpha helices. So you take 3 alpha helices, and this is what you do. You basically coil them around one another. So let me show you this. So here's one alpha helix. Okay. And then what you're going to do is take another alpha helix, and maybe it can be just after this one, and maybe I should actually put it this side, because this will be the portion that joins onto H alpha 1. Let's just rectify this. This is H alpha 1, H alpha 2 domain over here. Okay, so here's your first alpha helix, then you'll turn around and you'll have another alpha helix, and basically they'll be intertwined. Okay, so this one will intertwine with this other one, and this is why it's called a coil domain, because an alpha helix is already a coil, and now you're coiling the coils. So now we've coiled 2 alpha helices together, then you're going to come back with a third alpha helix, and this one's also going to intertwine around the other 2. Okay, and I know it's turning into a complete mess very quickly, but I hope this gets the principle across, that what you've got is these 3 alpha helices that are coiling around one another, and that's called a coil domain because the alpha helix is already a coil, and now you're coiling the coils, so it's a coil domain. Okay, so that's what you have in that, represented by that pink box there, these 3 coiled alpha helices basically. Okay, and the coil domain of a phospholipase C beta enzyme is specifically 3 alpha helices. Coil domains elsewhere can be different numbers of alpha helices, maybe just 2, maybe 4, etc. Okay, right, so the coil domain again is really important in the phospholipase C beta enzymes. It's believed that it's what actually binds the enzyme to the underside of the phospholipid bilayer. So let me now show this. So basically, you remember me telling you that with all phospholipase C enzymes, to activate a phospholipase C enzyme, there's 2 steps. Firstly, you have to get it to the membrane, and secondly, you have to actually activate it. You have to remove the XY linker from the catalytic site somehow. Okay, now, basically, phospholipase C beta enzymes can do the first step on themselves. They can attach to the membrane on themselves, by themselves, and then the alpha subunit of GQ/G11 proteins, all that does is then removes the XY linker from the catalytic site, and therefore does the second part of the activation. Okay, and it's the coil domain that's believed to be really important for attaching you to the underside of the cell membrane. So if I show this, okay, it's believed that there is this continuous equilibrium of the phospholipase C beta enzymes. Okay, they can continuously move between floating around in the cytoplasm and being attached to the underside of the phospholipid bilayer, and it's believed that it's the coil domain that attaches you to the underside of the phospholipid bilayer. So I'm going to redraw the coil domain now, like so. Okay, so it's now just going to be represented by these 3 alpha helices, and I know they're shown parallel, that's just to make the picture pretty. In reality, they'd be coiled around one another to make the coil domain. Okay, so here's the coil domain now bound to the underside of the phospholipid bilayer, and then the rest of the enzyme will be dangling into the cytoplasm like so. So here is the H alpha 1, and I'm sorry about writing it on a slant like this, H alpha 2, okay. Then what you'll have after that, I'll try and put it onto a more normal angle. Here you have the C2 domain, okay. Then you'll have, of course, the catalytic domain here, made up of the X box and the Y box, and forming this tin barrel-like structure with the catalytic site at the center there, occluded by the XY linker, which we'll have in vivid purple here. Okay. Then prior to the X box, you then have the 4 EF hand motifs here, like so, and then you have the plextrin homology domain here. Okay, and then prior to the plexogen homology domain, you have the amino terminus. Okay, so that's the structure of our phospholipase C beta enzyme attached to the underside of the phospholipid bilayer. So this is continuously moving between the cytoplasm and being attached to the underside of the phospholipid bilayer. So this is continuously breaking off, dropping into the cytoplasm, then it can bind to a new bit of phospholipid bilayer, etc. Okay, so there's an equilibrium basically. At any one time, some phospholipase C beta enzymes will be attached to the underside of the cell membrane, others will be in the cytoplasm. Okay, and this goes for all 4 phospholipase C beta enzymes, so I should have stressed this. All of what we're talking about applies for all 4 of them. Okay, so all of this structural stuff that we've been talking about, all of this moving between the cytoplasm and the plasma membrane, that's for all 4. Okay, right. So, at the moment it's still not active. Yes, it can move between the cytoplasm and being attached to the underside of the phospholipid bilayer, but, you know, at the moment the XY link is still occluding that, even though it's bound to the underside of the phospholipid bilayer. Okay, so, it's not going to be breaking down any PIP2 to diacylglycerol and IP3. Okay, and to activate the phospholipase C beta enzyme, the major way that they can be activated is through the alpha subunits of heterotrimeric G proteins, and this is the really famous way that phospholipase C beta enzymes can be activated. So let's now discuss G protein coupled receptors. Okay, so we'll come back to this picture in a moment. For now, let's have a little diversion and discuss G protein coupled receptors. Okay, right, so let's start with GPCRs then. So G protein coupled receptors. Okay, so G protein coupled receptors, they are a massive great family of proteins in humans. You have over 800 different G protein coupled receptors in the human genome. Okay, and all G protein coupled receptors share this same characteristic 7 membrane spanning alpha helices, which I'll draw like so. Okay, so this represents the cell membrane again, outer leaflet, inner leaflet. The amino terminus of the G protein coupled receptor is extracellularly, and the C terminus of the G protein coupled receptor is intracellularly. Okay, now, when the ligand binds to the G protein coupled receptor, it changes the conformation of the G protein coupled receptor intracellularly. It changes the conformation of these 3 intracellular loops here and the intracellular portions of the transmembrane alpha helices, so that this G protein coupled receptor can then interact with heterotrimeric G proteins. So let's now have a discussion of heterotrimeric G proteins. Okay, right. So heterotrimeric G proteins, as the name suggests, they are trimers, which means that they are made up of 3 separate proteins. And the heterotrimeric means that the 3 subunits that make up the trimer are all going to be different proteins. Okay, so let me just extend this membrane and then I'll show you the structure of a heterotrimeric G protein. Okay, so heterotrimeric G proteins consist of an alpha subunit, a beta subunit, and a gamma subunit. Okay, so I'll draw the alpha subunit here, so this is the alpha subunit, and then we'll have the beta subunit and the gamma subunit here and here. So this is the beta subunit and the gamma subunit. And for short, heterotrimeric G proteins are abbreviated as G for G protein, and then alpha, beta, gamma for the alpha, beta, and the gamma subunit. Okay, right. Now, heterotrimeric G proteins can be in 2 states. They can be in the on state, okay, in which the alpha subunit has GTP bound to it, okay, and in the on state, the alpha subunit with GTP bound to it is no longer bound to the beta-gamma complex, okay. So you have the alpha subunit with GTP bound to it, and then you also have the beta and gamma subunit which remain together and are called the beta-gamma complex. Okay, however, we're going to start off in the off state. Okay, and in the off state, the alpha subunit instead has GDP bound to it, and when it has GDP bound to it, it remains bound to the beta and the gamma subunit. Okay, so the whole thing remains as a trimer. So we'll start off in this off state where the alpha subunit has the GDP bound here, and therefore is attached to the beta gamma complex here. And because the beta and gamma subunit under no physiological circumstance ever come apart, they're called the beta gamma complex. So just to colour this in here, let's colour in the alpha subunit here in the red, Okay, the beta subunit in blue, and the gamma subunit here in green. Okay, right. The other thing to say is that the subunits of the heterotrimeric G proteins are attached to the plasma membrane via lipid moieties which have been attached to them. Okay, so the alpha subunit has a lipid moiety attached to it, which implants into the inner ethers of the phospholipid bilayer, and so does the gamma subunit. Okay, so the basic principle here is that onto the side of the protein, okay, you attach a lipid molecule. Okay, and in the case of alpha subunits, it's usually a palmitoyl group, okay, which is a 16-carbon fully saturated carboxylic acid, so a very long hydrophobic molecule. And this then implants into the inner ethers of the phospholipid bilayer and holds the alpha subunit attached to the underside of the cell membrane. Okay, now the principle is the same for the gamma subunit. It's a different type of lipid molecule. Instead, you attach either a farnesyl group or a geranyl group, but the principle is the same, that what you're doing is attaching a lipid molecule onto the gamma subunit, and this implants into the inner ethers of the phospholipid bilayer and holds the gamma subunit attached to the underside of the phospholipid bilayer. Okay, right. Now, there are loads of different types of heterotrimeric G proteins. Okay, so there are loads of different alpha subunits of heterotrimeric G proteins, there are loads of different beta subunits, and there are loads of different gamma subunits. Okay, we're not really going to care about the beta and the gamma subunits. Okay, for our purposes, we're just going to call them beta and gamma. We're not going to distinguish between all the different beta and all the different gamma subunits. Okay, what's important for us is the alpha subunit of the heterotrimeric G protein. Okay, and the alpha subunits that we're going to be interested in are in a family of alpha subunits known as the alpha Q/11 family of alpha subunits. Now, there are 4 members of the alpha Q/11 family of alpha subunits. The most famous alpha subunit in this family is the alpha Q subunit. The second most famous is the alpha 11 subunit. Then there are 2 that are much less famous, the alpha 14 subunit and the alpha 15/16 subunit. Now, this is one alpha subunit, okay, and to convince you of that, I'll just tell you the story behind how it became known as alpha 15/16. Okay, basically, the people working in mice discovered what they thought was a new alpha subunit of heterotrimeric G proteins, and they called it alpha 15. So this is the murine version, the mouse version. Okay, the people working in humans discovered what they thought was a new alpha subunit of heterotrimeric G proteins, and they called it alpha 16. Okay, later it transpired that alpha 15 in mice was the equivalent of alpha 16 in humans, and therefore there was a movement to have both of them renamed alpha 15/16. Okay, so that's why alpha 15/16 is called alpha 15/16. If you hear someone just talking about alpha 15, that means alpha 15/16. They just haven't accepted the new naming system. If you hear someone talking about alpha 16, again, that means they just haven't accepted the new naming system. Really, the ideal thing to call it is alpha 15/16. Okay, right. All 4 of these subunits actually do the same thing. They're all capable of activating phospholipase C enzymes of the beta family. So all 4 of the phospholipase C beta enzymes. Okay, alpha 11 is actually almost identical to alpha Q, and alpha 14 and alpha 15/16 are very similar. Okay, and they're all grouped into a family because they pretty much do the same thing. So even if you'd thought that alpha Q is the only one capable of doing it, in fact it's all of these 4 that are capable of activating phospholipase C enzymes of the beta family. Okay, and when people usually put alpha Q/11 here, it means any one of these alpha subunits in the alpha Q/11 family. Okay, and these are the heterotrimeric G proteins that we're going to be interested in, called GQ/11 heterotrimeric G proteins, because heterotrimeric G proteins, remember, are named according to which alpha subunit you have. So this means a heterotrimeric G protein where the alpha subunit is one of these alpha Q/11 family alpha subunits. Okay, right. So basically, G protein coupled receptors do not activate all heterotrimeric G proteins. G protein coupled receptors, once they've been activated by their ligand, are selective for certain types of heterotrimeric G proteins, determined by the alpha subunit, the beta subunit, and the gamma subunit. Okay, now we're not going to fuss about the fact that the beta-gamma subunit can confer which G protein coupled receptors are going to be able to activate the heterotrimeric G protein. As I say, we're just going to call that beta and that gamma. We're not going to talk about the different beta and the different gamma subunits. We're only going to worry about the alpha subunit. Okay, so there are certain G protein coupled receptors that are capable of activating heterotrimeric G proteins where the alpha subunit is in this alpha Q/11 family. Okay, and these are what are known as GQ/11 coupled G protein coupled receptors. Okay, so let me give you some examples of GQ/11 coupled G protein coupled receptors. Okay, so some very notable examples are the alpha-1 adrenoreceptor, and also the M3 muscarinic acetylcholine receptor. Okay, so those are notable examples which can activate GQ/11 heterotrimeric G proteins. Okay, right, now what happens then is that when the G protein coupled receptor binds its ligand, okay, and in the case of the alpha-1 adrenoreceptor that will be noradrenaline, in the case of the M3 muscarinic acetylcholine receptor that will be acetylcholine, okay, the G protein coupled receptor will change its conformation, which will produce this change in conformation on the intracellular aspect. It will now be able to bind heterotrimeric G proteins of the GQ/11 family, okay, where the alpha subunit is in this alpha Q/11 family, okay, and then what will happen is it will catalyze the release of the guanosine diphosphate from the alpha subunit, okay, and then a GTP molecule, guanosine triphosphate, will come from the cytoplasm, bind to that alpha subunit's guanine nucleotide binding site. It will cause a change in conformation in the alpha subunit that will firstly cause the beta-gamma subunit to break off from the alpha subunit. So this will now go off. It will also break off from the G protein coupled receptor, and it will go off on its own little adventure here. Okay, so here is our beta and our gamma subunit that is now cleaved off the alpha subunit. Now that the alpha subunit has got the GTP bound, so here's beta and here's gamma, okay, and now the alpha Q/11 with the GTP in, it will also change in conformation so much that it breaks off the activated G protein coupled receptor. So you'll also have the alpha Q/11 subunit now on its own, free from the G protein coupled receptor with the GTP bound to it. Okay, and both the alpha subunit with the GTP bound and the beta-gamma complex will remain attached to the underside of the phospholipid bilayer by these lipid moieties that attach them to the underside of the phospholipid bilayer, so by the palmitoyl group in the case of the alpha Q/11 subunit, and by the prenyl group or farnesyl or geranyl group in the case of the beta-gamma complex over there. Okay, so we've now got this activated alpha Q/11 subunit with GTP bound. What can it now do? Well, it can go and activate phospholipase C beta family enzymes. So basically, it activates phospholipase C beta enzymes that are already attached to the underside of the phospholipid bilayer. So remember I told you that the phospholipase C beta enzymes are in this equilibrium between being in the cytoplasm, floating around in the cytoplasm, and being attached to the underside of the phospholipid bilayer. The alpha Q/11 subunits are capable of activating the phospholipase C beta enzymes that are already attached to the underside of the phospholipid bilayer. Okay, so they're not capable of activating the ones in the cytoplasm, it's only the ones that are already attached to the underside of the phospholipid bilayer. And basically what it's going to do is, if I show the alpha Q/11 subunit here, so here it is coming along, attached to the underside of the cell membrane, like so, with its GTP bound. What it's now going to do is it binds to two major regions. Whoops, miscolored this in. It binds to two major regions on the phospholipase C beta enzyme. Firstly, it binds to this H-alpha-1, H-alpha-2 region, this helix-turn-helix motif. In addition, it also binds to our coiled coil domain here, okay? And what it ends up doing is it ends up reorienting the phospholipase C beta enzyme. Specifically, it ends up bringing this tin barrel-like domain up to the underside of the phospholipid bilayer. Okay, and when you bring this up to the underside of the phospholipid bilayer, that, as I described in my initial discussion of the catalytic domain, causes the electrostatic repulsion of the XY linker here. Okay, because the XY linker has the negative charge on it. Okay. It then gets repelled by the negative charge of the phospholipid bilayer, and that causes the XY linker to move out of this catalytic site here, okay, which then opens this catalytic site and makes it available now for the phosphatidylinositol 4,5-bisphosphate molecule to stick its head into the catalytic site. Okay, so the alpha Q/11 isn't involved in recruiting the enzyme to the plasma membrane, it's involved in activating the enzyme once it has been attached to the underside of the plasma membrane, okay, by just reorienting the enzyme and allowing electrostatic repulsion between the XY linker and the underside of the phospholipid bilayer to remove the XY linker from the catalytic site. Okay, and that then is now going to leave us with an activated phospholipase C beta enzyme, which will convert PI45P2 into diacylglycerol and IP3, and the diacylglycerol and IP3 can then go off and have signaling roles such as creating a calcium signal and activating protein kinase C enzymes. Okay, right, so that is the most famous example of the signaling pathway which phospholipase C enzymes are involved in. In the next video, what we'll do is continue our discussion of phospholipase C beta enzymes, okay, by discussing how certain phospholipase C beta enzymes, specifically phospholipase C beta 2, can be activated by G beta gamma subunits. And then what we'll move on to is the phospholipase C gamma enzymes. But before we just end this video, I would like to include one last little nugget of information, okay, and that's to tell you that the phospholipase C beta enzymes are not all equally well activated by GQ/11 alpha subunits, okay. This mechanism of activation is best for beta 1 and beta 3. Okay, so these ones, phospholipase C beta 1 and phospholipase C beta 3, they are absolutely brilliantly activated by alpha Q/11 subunits with GTP bound. It's not as good for activating beta 2, and it's much, much worse at activating beta 4. So these two are fantastically activated by alpha Q/11 with GTP bound. Beta 2 is reasonably activated. Beta 4 isn't very activated at all, okay? So beta 1 and beta 3 are the main ones that can be activated by this mechanism, but they can all be activated to some extent by alpha Q/11 with GTP bound. Okay, right, so in the next video we'll go on to the G beta gamma regulation of phospholipase C beta enzymes, and then we'll move on to phospholipase C gamma enzymes and how they can be activated by receptor tyrosine kinases.